All right. Thank you for the nice words, and certainly a great honor for me to be here today talking about my work. And as Georgia Love said, I specialize on the study of microbotanical remains, especially on phytoliths and starch grains. And you see. Okay, so certainly it's a big topic, and there's a lot of things to say. I decided to use this lecture as an introduction to microbotanical research in archaeology. And for this task, I decided to structure the lecture in 4 blocks. First, to explaining what microbotanical remains are, what phytoliths and starch grains are, how they form, what are their characteristics, and what information can provide, and how they have been implemented in archaeological research, and in this case, how they have been used to research plant use, foodway practices, and we also mentioned some of the processes regarding sampling and laboratory protocols. Then I would have a practical example on how microbotanical analysis are implemented in archaeological research based on my work for my PhD thesis at Chateaubriand, and present, for example, how they have helped to investigate foodway practices and household activities. And finally, I'm going to conclude with a discussion about the most relevant findings of this of the work on Chateaubriand, and some notes and final remarks about further lines of research in botanical analysis in Western Asia. So first, let's start by explaining what phytoliths are. Phytoliths are basically silica bodies that they're produced by the deposition of soluble silica in the intercellular plant tissues, where it's solidified. In this sense, the shape and the size of phytolith will be determined by the location or the plant tissue accumulating the silica, or the cell accumulating silica. And this solidification pattern is known as incidental solidification. And once it's important to mention that not all plants produce phytoliths, but they're highly produced in grasses specifically, and you can find them in most plant tissues, for example, leaves, stems, flowering parts, or inflorescence, root and fruit. Then once plants decay and plants enter the soil, we see how the phytolith cycle starts over. So what information can provide? Well, first, the classification and identification of phytoliths is standardized. For this, we use the International Code of Phytolith Nomenclature, the 2 versions, the 1.0 and the 0.02. Once we are able to identify correctly the phytoliths, they can be useful to trace the phytoliths into the plant tissue where they form, for example, cone leaves or inflorescent parts. And in some cases, it might be possible to identify plants to the family, genus, or even species in the case of using morphometric analysis. And one of the main advantages of phytolith is that since they are inorganic, they can survive for thousands of years under stable conditions in sediments, which allow us to have another proxy for the recognition of vegetable resources that might otherwise not be observed in the archaeobotanical record due to preservation biases. So starch grains, on the other hand, is a complex carbohydrate. It's composed of amylose and amylopectin, which are 2 glucose polymers, and that form this insoluble semicrystalline structure that we call starch grain or granules. The formation of the starch from the hilum or the center. Let me see, from the center, where association of chains of amylose and amylopectin start to develop in bands, forming the structure. Let's see here. These bands, and this is called lamellae. And one of the characteristics of the starch grains that they have an optical property on the cross-polarized light, which is called bifurcance, that creates the so-called extinction cross or Maltese cross. And this is very important because this allows us to identify starch grain within the sediment. And there are actually 2 categories of starch grain. They are transitory starch and storage starch. The transitory starch forms in photosynthetic tissues as a form of energy that can be used right away for the plants or later during the day to sustain metabolic functions in the plants. And then we have storage starch, which is the one that we are interested. It forms in specialized organelles called amyloplasts for long-term storage in special organs, for example, in above-ground stems, fruits, seeds, underground storage organ, which in this category we include tubers, root, rhizomes, and all these storage organs that we can find on the ground. And the function of a storage starch is to supply energy, in the case for seeds, to allow the germination and the initial growth. And in the case of underground storage organs or geophytes, the function of this kind of a starch is to maintain the metabolic functions in plants during moments of stress, for example, during winter, when the plants begin dormancy and start blooming later on spring. And this is the kind of a starch that has been targeted by humans for millennia as a food source, and nowadays it contributes to the caloric intake of many people around the world. Basically, wheat and barley, this is pure starch. So what information can provide? Well, knowing where and how a starch forms certainly has some important implications for understanding the plants uses and the pathways of plant resources, of starch-rich resources into the archaeological record. And for the identification of a starch root, we follow the International Code for Starch Nomenclature, and we consider several factors, for example, size, length, the shape in 2D, 3D, other features like pilum, which was mentioned for the center, facial facets, border, lamellae, and also the shape or the form of the finching cross. All these different characteristics are valuable at the time of identifying the starch to different origin. And another property of starchment is that certain activities, certain anthropic activities can leave traces in the starchment morphology. For example, we have here native starchment, which basically is modified wheat starch, and we see how it's been transformed by yeast, which can occur during fermentation, and by heat, in this case, parching. So in this case, depending on the context, starchment can be also very helpful to understand different complex processes on culinary practices. And so how phytoliths and starchment have been implemented in archaeological research? Well, they have simply many applications. Phytoliths can be used for environmental studies, for ecological studies, but today I will focus on how they have been used to study foodway practices, and in particular with this example in crop processing sequence, starting from harvesting, processing, cooking, and eating. In this sense, phytoliths can provide information about plant exploitation, the production of plant-based craft like biscuitry, and as I said, the first steps of crop processing where the non-edible part, the chaff, the leaf, are removed, as we can see here. And once we move further in the crop processing sequence, we see how starchment tend to be more informative for further steps in the sequence, identify the presence of starch-rich resources that normally do not survive. And for example, recovered from the grinding tools or utensils used during cooking activities. However, since they can provide information from both sides of the sequence, certainly using phytoliths and starchment together have great advantage, since they complement each other by revealing the multiple steps involved in plant processing sequence and the presence of a starch-rich resources through the whole sequence. So regarding the archaeological application and microbotanical analysis, despite being susceptible to biological chemical degradation, starchments like phytoliths can survive in archaeological sediment for thousands of years, and they can provide us insight or a record of the artifact life history or the last use of the artifacts, and they can be recovered from residues originating from material culture involving plant processing artifacts, cooking utensils, and even human remains, more specifically, grinding tools, dental calculus, coprolites, cooking vessel, food cross, and even the activities where these plant processing activities are taking place, so the activity areas where the activities are taking place. So once we recover the samples, we have to move to the lab and where we proceed to the recovery of microbotanical remains from the sediments. In this sense, there are multiple protocols for this task, and they may change depending on the proxy that we're targeting. For example, some are specific for starch, some others are specific for phytoliths. It also depends on the kind of residue, if we are studying dental calculus, but in general, all of the protocol has a main goal, which is extract the microbotanical remain from the soil matrix or from the sediments. For this task, I follow a combined protocol to extract both starch grains and phytoliths from the same sample, and it can take a little bit more than 1 hour. But just a summary of the protocol, we start with sample preparation, which label and weighting. Then we proceed to clay defiltration, which basically loosens the clays to allow the starch grains to be free for the soil matrix. And we recover the starch grain first by flotation using a heavy liquid in order to avoid the exposure of the starch grain to other heavy chemical reactions that may occur later during the phytoliths procedure that can cause heavy damage to the starch grain. And this is something that certainly we want to avoid. Once we have the starch grains safe, we proceed to remove the carbonates and organic matter using hydrogen peroxide and hydrochloric acid. And again, we then remove the phytoliths from the remaining sediments using flotation and heavy liquid. And then samples are ready to be studied under a microscope, or we can store samples for later analysis in the future. I invite you all to visit this protocol in protocols.io, where you can make comments on it, so we can keep improving it and making it more user-friendly, which is the main goal. So after we have finally the samples ready, we have to move to the procedure regarding microscopic analysis. In this sense, the microscopic procedure for phytoliths and starch grains are similar, but they have major difference. First, starting from the mounting, slides for phytoliths are mounted using a permanent mounting medium. Then they are observed under the microscope at 400 magnification up to reaching 250 single phytoliths, which is a statistical account that we know is representative for the whole sample. Then we proceed with the data analysis, and in this part, we can also consult the reference material in order to identify phytoliths that might be unknown to us, and we can use reference collections from online or plants that we can have in the lab. And finally, if we have the chance to find silica skeletons or phytoliths in anatomical connection, we can also perform morphometric analysis in order to know if these phytoliths are coming from one of the species of wheat or barley. And this can also be done with some pineapple grasses or millets, for example. Now for starch grains, basically it's very similar, but in this case, slides are prepared using a mobile medium. In this case, we use glycerin in order to rotate and move the starch grain to be able to record all these features that I mentioned before. These are also observed, and this time on the polarized light, in order to be able to identify correctly the starch grain by the extinction cross. And once we are sure that this is certainly a starch grain, we start with the recording of the different features, the lamellae, the ilum, the extinction cross. And again, we can consult reference collection. This is part of my personal reference collection of all the ground storage organs of the wild plants in order to refine the identification when possible. Sometimes we cannot tell exactly where it's coming from, but certainly reference collection can help a lot in this part. And then we proceed with the data analysis. So now I will proceed to demonstrate with a practical example how all these protocols, all these processes are put in practice while showing some examples from Çatalhöyük. So let's introduce first the site. So Çatalhöyük is a 9,000 years old farming community located in the Konya Plain, Turkey, and it was discovered in 1958 by James Mellor, who started excavation later in the early 1960s, uncovering a large Neolithic settlement with a remarkable state of preservation of mud brick structures and material culture. Later on, in 1993, the Chateaubriand research project was launched under the direction of Ian Hodder, lasting for 25 years of continuous excavations. Well, Chateaubriand is actually composed of two mounds, east and west, and most of the archaeological research and the most famous is the east mound, where the excavation have been focused in two areas, the north and south. In the north area, excavations focus on exposing spatially the sediment to understand the spatial distribution of different houses during the middle occupation period, and is known as the classical Chateaubriand. The south area is chronologically earlier, and here the excavation focus on trying to reach the lowest part of the earliest layers to understand how the site was developed. And regarding the urban plan, Chateaubriand can be considered one of the densely agglomerated mega sites with multiple buildings and blocks. They were very densely packed and entered to the roof. And a very interesting characteristic of Chateaubriand and other sites in this region is that the internal spatial arrangement of the domestic space was very standardized. Basically, in all the houses in the north part, we can find benches, we can find platforms and other structures. And in the south is where we can find the areas for cooking activities. For example, hearths, ovens, ladder entrance were always on the south. This division of space was very, very well standardized in all houses. This is a pattern that all houses follow. So what we know about plant use at Chateaubriand, thanks to previous approaches to plant processing activities, these have been basically based on the recovery of microbotanical remains. In this sense, Chateaubriand has produced by far the largest archaeobotanical assemblage for any other Neolithic settlement in Western Asia, with many samples. And thanks to the study of this large assemblage, we know that the farming economy of the people from Chateaubriand was based on the cultivation of bloom, free-threading cereals, legumes, and the incorporation of wild fruits and nuts. However, we have to be aware that microbotanical assemblage may actually represent a small portion of the whole spectrum of economically important plants, because not all plants preserved by sharing. Sharing usually occurs by accident. There are other causes that may produce, that may avoid the incorporation of other plants in the archaeobotanical assemblage. For example, raw consumption, processing, or there are some plants that simply are not prone. They don't preserve by sharing. This occurs with leaves, some fruits, underground storage organs. And all these components, we always hypothesize as we know they must be there, but because we don't have evidence in the botanical record, these are usually known as the missing plants or the undetectable components of diets in microbotanical records. So other approaches to understand plant processing activities at the household level at Chateaubriand have relied on understanding, at least at the macro scale, to understanding the distribution of charring remains and grinding tools in different houses. For example, this is building 77, where we can see the location, the different grinding tools in the house. And there have been many studies based on the distribution of this set of artifacts and remains that have been used to approach how plant processing activities were performed on a daily basis at the site. However, we have to remember that many of these tools are mobile, and also char remains can be moved around. So this always raises a question about how accurately the observed distribution of microbotanical remains and artifacts are actually representing everyday activities in the past. In addition, this is not very common actually at Chateaubriand. Chateaubriand is mostly, houses are mostly clean, devoid of artifacts, which means that even though we find this kind of arrangement, we always have to be aware that maybe this is just the last location of the artifact, but how we know this happens every time, this is a big question. From microscopic scale, we have to be aware that the plant processing activities involved in these activities, and even the activity areas around where the activity is taking place, can actually contain microbotanical remains like phytoliths and starch grains, despite there's any other visible a signal for these plant resources in the archaeological record. And despite that, certainly microbotanical analysis has been implemented at Chattahoyuk before. Its application specifically for researching plant processing artifacts, grinding tools, and other implements, and house floors to understand the distribution of plant processing activities and the organization of the living space regarding plant processing tasks. This kind of application have been preliminary in general. This means that there's a lot of work to do regarding this aspect. So based on this awaiting potential, we decided to perform an integrated microbotanical approach based on an artifactual and spatial approach. And for this task, we focus on case study, in the case study building 80. I have to mention that the study is way more larger. We include more artifacts, we include other buildings, but today I just want to focus on building 80. Building 80 is located in the south area. It was inhabited between 6,700, 6,500 BC. And one of the most interesting features of this building is the presence of a fixed grinding installation in the central space of this building that was there through multiple occupation phases. And in some way, it may have dictated or influenced what kind of activities were taking place in this area of the house. Another interesting characteristic of this building is that it lacked an oven, like most of the houses at Chattahoyuk. The only combustion feature in this house was this heart, which always bring question to how the inhabitants of the house covered the food-related needs during the time there was no oven in this building, but when the grinding installation was in use. So due to this characteristic, building 80 is actually a great example to perform an integrated approach in combining the artifacts involved in plant processing activities and to look at the remaining surface around this the whole building. Well, the artifactual approach consisted on recovering sediment samples recovered from the crack and porous surface of the fixed cairn. These sediment samples were later exposed to the protocol of double extraction of phytoliths and starch-rich, the protocol that I mentioned before, and then the data analysis with the quantification of microbotanical remains and to identify them into taxonomic or anatomical level whenever it was possible. The spatial approach was similar, but however, here the target was to identify inputs on microbotanical remains within the house that could be traced back to or may originate from the repetition of processing activity, plant processing activities in the same location of the house. For this task, we conducted a grid sampling to the whole house every 50 centimeters when possible, always avoiding bioturbation and other things that may actually give us an incorrect sample. And once at the end, we covered 83 sediment samples that were georeferenced and exposed again to the double extraction of phytoliths and starch-rich by flotation, and again were classified into anatomical taxonomical categories whenever it was possible. However, here we need to take the analysis to a step forward, because one of the main problems is that phytoliths are everywhere, especially in a site like Chattahoyuk, that we have mud bricks, we have plasters, and you use crop byproducts to produce this kind of built environment. So one of the biggest questions is how we are sure that these inputs are actually representing or originating from plant processing activity, and it's not just phytoliths that are just part of the built environment. So for this task, I decided to follow a statistical methods, in this case, kriging and inverse distance weight, which is a method developed through minings, which are actually used for a similar problem. You know, in mining industry, they want to know where in the whole mine you have more accumulation of whatever minerals you're targeting. So here we had the same problem. We want to know where in the house we have more accumulation of phytoliths. So for that reason, I decided to use this method. And spatial prediction method we're creating our studio, and the map show areas of enrichment of microbotanical remains in red and areas of depletion in blue. Then these spatial prediction maps were interposed in QGIS in the building frame in order to have a better understanding on how this concentration relate with the internal arrangement of the building. So now let's go for the result. And regarding the result from the fixed binding installation, it was very surprising to see a great amount of phytoliths with all samples reaching 200 single count, so 250 count of single phytoliths, and it was dominated by inflorescence of grasses, in this case wheat, barley. This is already telling us that this artifact was indeed involved in plant processing activities. But however, it was also surprising to see that we only recovered 11 starch grains, which is certainly low and unexpected considering the great evidence for plant processing activities that is already telling us from the phytoliths. I will discuss this further later. Regarding the phytoliths from house floors, they were very abundant as well, with a total of 20,750 single phytoliths identified. Obviously, a work of a lot of months under the microscope in the lab. Then these phytoliths were classified into broader taxonomic and anatomical categories in order first to make data more manageable and in order to create better categories to be implemented later for the spatial analysis. And these categories were grass inflorescence, grass culm leaves, panicle grasses, and Ericaceae. Ericaceae are palms. We can see from the spatial analysis that once again, phytoliths from grass inflorescence were associated or concentrated next to the fixed binding installation, and also on the heart, which is also telling us from the spatial analysis that indeed there were certain plant processing activities involving cereals in this area of the house. Panicle grasses were found next to the heart, but one of the most remarkable findings is the presence of Ericaceae phytoliths from palms. Why? Because palms are not local for Central Anatolia. So this is certainly like an intrusion or exogenous plant that was present in Çatalhöyük, and I'm only presenting this building, but they were found in other contexts as well. And this is a very interesting find. Regarding the starch grains, they were also very abundant for a total of 1,750 recorded starch grains, which were later classified into 21 typologies based on similarities and distinctions, and following the same method as phytoliths, they were classified into broader taxonomic groups such as cultivars representing cereals and legumes, and wild resources, in this case wild grasses and geophytes. Here we can see that the starch grains or cereal starching are actually concentrated away from the grinding installation, but mostly closer to the heart. Legumes were also located in this location, away from the grinding installation. But interesting was to see also evidence from panicle grasses, also located next to the heart. But certainly one of the biggest surprises from the analysis of starching was the presence of a lot of starching from underground storage organs, from roots. They are represented by 10 typologies. There you go. So by 10 typologies, and this is the first time many of these plants have been identified at the site, which is already just by finding all this presence of underground storage organs telling us a lot of information that we did not know from before. So now it's time to merge all this data produced by the grinding tool, the phytoliths from the house floor, and from all the data, and to try to see how this information can actually tell us new aspect of plants use at Çatalhöyük. So thanks to the starch grain, we were able to broaden the spectrum of plants used at Çatalhöyük. In this case, I want to hesitate again the presence of underground storage organs, where after contrasting starch grains found on the house floor with reference material, we were able to identify them as originating from the rhizomes or the underground storage organ of Illyriaceae or Liliaceae, basically the bulbs or lilies For example, tulips belong to this same families, but at the moment we can only identify them into the family level. We cannot go further, but this is certainly a very interesting find because this kind of resources never survive in the macrobotanical record by sharing. And another interesting find was that after constructing the distribution of how these different categories for underground storage organs, starch grains were distributed within the house, it was noticed that some group of starch grains from underground storage organs had different patterns that perhaps is telling us about the different use of these plants in different areas of the house. For example, we have group A associated to the grinding installation was here, that can be interpreted as a processing activity area. We have group B located also in the north, but in the storage area. And most interesting, at least for me, is those that are located next to the heart. Actually, this is the main heart. This is a fire spot, and it perhaps will be telling us about the intentional heat treatment or cooking of underground storage organs in this location. Certainly, roasting, baking, or drying tubers may help to eliminate toxins, because some of these plants can be toxic. Can enhance peeling, helps to process, and also the digestibility, because we're cooking the starch. And as I mentioned before, despite we are only able to identify them to the family level, the fact that we see different distribution of different typologies within different areas of the house is telling us already that we are dealing with different, probably more than one for sure, species or geophytes that were somehow used for different purposes at Çatalhöyük. And again, these resources are very, very informative because some of them has a restricted annual life cycles, and they can be used to have insight about seasonal plant procurement tasks. For example, we know that thanks to ethnographic data from Turkey, that some Iridaceae and Iridaceae geophyte species are collected in spring when the plants are blooming. For example, this is from a study by J.C. Leal, 1929, where these tubers are identified when they're blooming and they're consumed raw, or they are also consumed in other dishes. And this is particularly important because, as I mentioned before, some of these plants can be toxic. So we need to be able to identify them correctly by the floral part in order to make sure we're not making a mistake. And yes, and in this sense, this is another line of evidence that is telling us the presence of this plant. And also, if we think about it, if they're blooming or they're collecting in the end of the spring, that might actually coincide with the time of the year where the people from Çatalhöyük might be running low in supplies by the end of winter, and maybe the collection of these wild resources help or were a good complement in this time of the period where perhaps the supplies were running low. Either way, we know that if they were collected for sure in spring. And again, talking about the presence of wild plants, thanks to the combined information for phytoliths and starch grains, we were able to refine the identification of some of the wild grasses. For example, some of the starch grains, specifically two typologies, we were able to classify them into Setaia retigulata and Sorghum jalapense, where we can see they're very similar. They have similar morphometric characteristics and measurements. And by doing morphometric analysis on these phytoliths with this undulated pattern and comparing them with the phytolith produced in the floral part of Setaia retigulata, we were able also to match the phytolith with this plant. So this is a great example to demonstrate how well phytoliths and starch grains combine each other. When merging these two lines of evidence, we can actually have more accurate identification of the plant resources. So this is why it's very good to do the both analysis at the same time. And again, I want to mention again that both phytoliths and starch grains were located basically in the same location in the house, where we also find the cultivars. So perhaps this distribution is telling us already that these plants were indeed intentionally used in this area of the house, whether for food or any other proposes that at the moment we cannot tell. But these plants were there for sure, and this is something very interesting because, as I said before, these plants were not identified in the Ecobotanical record before. And another interesting insight that we can get by combining all the phytoliths, the starch data from the house floor, from the grinding installation, is that strong evidence for house-based post-processing activities or processes. In this case, we see by the concentration of cereal inflorescence around this grinding installation that indeed cereal processing activities were taking place. We remember that the artifact was actually full of starch grains from cereal inflorescence as well, but the lack of a starch grain both on the artifact and in this area of the house from cereals is telling us that yes, it was used for plant processing, but perhaps during the first steps where we clean the cereals or where the non-edible part where the phytoliths are located. For example, activities like the husking or threshing. We cannot tell for sure, but what the evidence is telling us for sure is that fine grinding was not taking place here, and this artifact was not used, at least at the moment, for making fine flour or something like that. And we can see later the concentration exactly in the heart, but it's also telling us the use of the crop byproduct for fuel. All right, so I want to bring back the question or the issue with the palm phytoliths, because what they are doing there, no? So there are actually two options for the origin of palms phytoliths at Chateaubriand. One is in the Levant and the Aegean coast, where we can find Phoenix dactylifera, which is date palm, and there's also another local palm in the Aegean coast called Phoenix theopraxi. This one, we know that it has been used since antiquity for basketry. Actually, in other sites in the Mediterranean where we find where phytoliths from palms are found, these are always associated with contexts related to storage activities or basketry. And so for that reason, when we find phytoliths from palms, it's always associated with the presence of basketry or palm-based crafts. So perhaps this is telling us about exchange network, perhaps from the Aegean coast, where this kind of craft might be arriving at Chateaubriand. And the location of this concentration is very interesting as well, because here's where we can find the ladder bench. You know, the house was entered to here. So this location here perhaps is telling about a dynamic image where perhaps by entering or getting out of the house, this plant-based craft were located in this location, and through time, through the repeated location of this artifact in these areas, when we see how phytoliths are free from the plant, and they became integrated into the sediment. You know, this is something that doesn't occur by once. It's something that occurs through time. But it's a very interesting find, and it's also worth mentioning because it's telling us about the presence of palm-based craft at Chateaubriand. Basketry or other kind of craft have been found before, but never from palms. And this is a contribution for phytolith analysis. So now getting to the end of this lecture, I would like to make some conclusion and final remarks about to demonstrate how microbotanical analysis have proved to be a valuable tool for researching complex viticultural practice and past libraries at Chateaubriand. From the study of Chateaubriand, the identification of the analysis of microbotanical remains has shown complex use of wild resources, shown by the identification of a wide range of wild resources such as geophytes, wild grasses, whose contribution of past diets or past plants use hasn't been always hypothesized, because we know they must be present, but because the lack of evidence, the use of this artifact has been rarely firmly established. And we can also get insight about the seasonal use of plants at Chateaubriand, where microbotanical remains can inform about the seasonal procurement of plants and provide a dynamic image of the different plant-related tasks that once were formed part of the inhabitants' lifeways. And finally, I think we have demonstrated that microbotanical analysis can also be used to address complex questions like household organization, where the microbotanical data demonstrate that plant processing activities at Chateauroux in these houses were substantially arranged in the living spaces, and this formed part of the daily activities that once were taking place in these houses. And but certainly there's always areas for improvement, like always, and where, in particular speaking, in the case of Western Asia, despite the growing implementation of microbotanical analysis for researching past foodways and plants used in this region of the world, this approximation remains marginal compared to other regions where phytoliths and starches are more recurrently used. This lack of applicability may certainly relate to some specific challenge, among them, time and specialized equipment. Time constraints and the need for specialized equipment indeed may cause some lag and hinder the further application of microbotanical analysis in comparison to other approaches in archaeological research or in archaeobotanical research that can be done on site. However, as I have shown today, despite this constraint, microbotanical analysis can still provide novel information, even when we think there's nothing more to add. Like in a site like Chateauroux, with this great amount of evidence for plants use, still there's something more to add and more beneath the eye that microbotanical analysis can help us to understand. Contamination is another challenge. Well, contamination from modern starch-rich is a significant concern in ancient starch research, and actually, yes, indeed, incorrect handling can lead to substantial loss of time, resources, but for me, most importantly, for time, because we only able to see if this is contaminated once we do the sampling, once we do the laboratory protocol, 2 or 3 months, and then looking at the microscope and see the sample is contaminated. So certainly, it's a challenge, but although there are various protocols to avoid this kind of contamination of material, I think it's always advisable to have an expert on site to make sure that everything is done correctly and to not lose time and resources in contaminated samples. And finally, comparative material. I think this is also an area that needs further development, where certain microbotanical reference collections are laborious, but in the case of Western Asia, the prolific ethnobotanical literature produced by local researchers, for example, in Turkey, creates a strong foundation for the development of a reference collection that can also help us to have a better understanding of how these plants can be used. And I firmly believe that once we're able to expand reference collection, perhaps there will be a moment where we can go further down into the taxonomic classification of many of these wild plants, perhaps to move from family to genus. But this is something that only time will tell. And to conclude, I think despite all this challenge and constraint, there's a promising future for ancient phytoliths and starch-rich analysis in this region of the world. And it's hoped that this lecture may help to envision or make you curious about the application of microbotanical analysis to continue expanding our understanding of human-plants interaction in the past. And thank you to all of you. Here's my email in case someone wants to have more questions. And I will leave this reference here with some of the works mentioned in this lecture and some of the pictures that I may use of them. Thank you.